Hello everybody and welcome to my presentation on Depside Depsidone synthesis in lichenized fungi, insights from Pseudevenia furfuracea. Depsides and depsidones are polyphenolic compounds, and in depside, the phenolic rings are joined together by the ester bond, whereas in depsidone, apart from the ester bond joining the rings, there is an additional ether bond. What we know about the synthesis of these compounds or natural product synthesis in fungi in general is that the genes synthesizing these compounds are organized linearly into clusters called as biosynthetic gene clusters. These clusters have a core gene synthesizing the backbone of the compound and other additional genes. In case of depside-depsidone synthesis, the core gene is a non-reducing PKS which synthesizes the phenolic rings and joins them together via ester bond. For the synthesis of depsidone, another gene, cytochrome P450, is required, which converts the depside into depsidone via formation of ether bond. What we do not know about these compounds is that an organism may have up to 50 gene clusters, whereas the identified compounds from an organism is in general up to 5. And in most of the cases, a compound cannot be associated with a biosynthetic gene cluster. So most of the times it is not known which gene synthesize the compound. Another knowledge gap which popped up only recently is when the PKS from Pseudevernia furfuracea putatively synthesizing olivetoric acid was heterologously expressed, it produced lechenoric acid. Lechenoric acid is similar to olivetoric acid in the sense that they both have two phenolic rings joined by ester bond, except that in the lechenoric acid, the carbon side chain is missing. Production of lechenoric acid by a Pseudevernia furfuracea PKS is strange because lechenoric acid is not synthesized by Pseudevernia furfuracea. This raises the question if in the lichen, which would be Pseudevernia furfuracea in this case, other genes work together with PKSs to produce the depside or the metabolite. In this regard, we do not know anything from lichenized fungi, but from non-lichenized fungi, it is known that specific fatty acid synthases work together with PKSs for metabolite production. So here is the fatty acid synthase dimer. These FASs produce the carbon chains and deliver it to the PKS. PKS then synthesizes the phenolic rings. In the resulting metabolite, the rings are contributed by the PKS and the carbon side chain by the FAS. We propose that a similar mechanism exists in lichenized fungi as well, where FASs work together with non-reducing polyketide synthesis to produce the metabolite. Transfer of only or heterologous expression of only non-reducing PKSs therefore results in the production of a different metabolite, which in this case or in the experiment is the lechenoric acid, which is not having side chains. To fulfill this knowledge gap, I chose as my study system Pseudevenia furfuracea, which constitutes two naturally occurring chemotypes, the depside olivetoric acid producer and the depsidone physodic acid producer. These two compounds are basically the same, except that olivetoric acid is a depside and physodic acid is the depsidone. With this, I come to my questions. I was interested in identifying the clusters likely responsible for the production of depside and depsidone in Pseudevenia furfuracea. And I was interested in knowing if there are homologs of fungal fatty acid synthesis in lichenized fungi. To answer these questions, we sequenced the high-quality PacBio genomes of both chemotypes and identified biosynthetic gene clusters. In total, there were 55 gene clusters present, and out of which 8 clusters had a non-reducing PKS as the core gene. In order to find the putative olivetoric acid PKS, I added the sequences of Pseudevenia furfuracea non-reducing PKSs to a recently published PKS dataset and generated a maximum likelihood tree. In this tree, the PKSs are categorized into several groups depending upon the compounds they synthesize. For instance, these are usneic acid synthesizing PKSs, these are atronin synthesizing PKS, and so on. Out of 8 Pseudevernia furfuracea PKSs, 2 grouped with Depside synthesizing PKSs. Here. To select the putative olivetoric PKS between these two, I then use the following 2 criteria. I zoomed into the PKS to look for the expected domain structure. So the putative olivetoric PKS should have 2 ACPs, which are a prerequisite for synthesizing and dimerization of the phenolic rings. Next, I looked into the other genes present in the cluster. For the production of depsidone, for instance, it would be required that the cluster has a cytochrome P450. Using these criteria, I ended up with 1 cluster, which is our candidate cluster for olivetoric PKS. This cluster has the NR-PKS with 2 ACPs and a cytochrome P450 gene. So summarizing, my first results are that I identified the likely cluster for Depside depsidone synthesis in Pseudevernia furfuracea based on phylogenetic evidence, PKS domain architecture, and cluster gene composition. This cluster is highly syntenic between the 2 chemotypes. It has 10 genes, including a core and non-reducing PKS and a cytochrome P450. My second result is that I found the homologs of fungal fatty acid synthesis in Pseudevernia furfuracea. FAS constitute a multi-enzyme complex which delivers the carbon side chains to the PKS to start the metabolite synthesis. These side chains are then embedded as the SL side chains in the resulting metabolite. Summarizing, we propose the following mechanism for the Depside depsidone synthesis. Fatty acyl synthesize and deliver the carbon side chains to the PKS. Polyketide synthesis then take place on the 2 adjacent ACPs in the PKS, followed by cyclization and ring formation. The 2 rings are then joined together by the ester bond, and it's released, and the resulting Depside is released from the PKS. Depending upon if the organism is the depsidone producer, the cytochrome P450 then converts the Depside into the depsidone. So what are we adding into the known mechanism of Depside depsidone synthesis? It is already known that the non-reducing PKSs are capable of producing the Depside from a simple precursor, and the cytochrome P450 is required to convert it into the depsidone. We are adding that for the synthesis of Depsides that have acyl carbon side chains, fatty acyl synthesis work together with PKSs to produce the Depside. In conclusion, to conclude, I found that the same clusters produce the Depside and the depsidones in Pseudevernia furfuracea. And I found the homologs of fungal fatty acyl synthesis in lichenized fungi. We propose that fatty acyl synthesis take part in the metabolite synthesis in lichenized fungi as well as is known from non-lichenized fungi. With this, I would like to thank you all for your attention, and I would be happy to take questions. Thank you very much.